In this session, we are dealing with the structure of nucleic acids, nucleotide and nucleoside. This is very important concept from the chapter biomolecule in your grade 12 chemistry. Okay, so nucleic acid. We studied two important nucleic acids in biology. They are DNA and we have RNA. What is meant by DNA, dear all? DNA stands for deoxyribonucleic acid. What is that? Deoxyribonucleic acid. And RNA stands for what? Ribonucleic acid. Let us discuss detail about what is this species, how they are forming, and what are nucleic acid, etc. Okay, so dear all, the main component in DNA and RNA are, okay, a DNA and RNA, when you hydrolyze, okay, it having mainly three components. The DNA and RNA, when you hydrolyze, dissolve in water, it will mainly gives three components. So there are three main components in a DNA and RNA. What are the three main components? Dear all, DNA and RNA contain first part is called a pentose sugar. Then nitrogenous base, or we can say nitrogen contained base. Nitrogenous base is there, nitrogenous base. And we have phosphoric acid. Phosphoric acid is the formula H3PO4, that is called phosphoric acid. Okay, these are the main component in DNA and RNA. So DNA and RNA on hydrolysis, we will get pentose sugar, nitrogen contained base, nitrogenous base, and the third one is called which one? Phosphoric acid, the formula H3PO4. Dear all, the pentose sugar part in DNA and RNA are different. Nitrogenous bases, nitrogen contained bases are in DNA and RNA are different. And phosphoric acid, that's the one common. Phosphoric acid H3PO4, we can draw the structure like P double bond O, OH, OH, OH. See, 3 hydrogen, H3PO4. This is the structure. Okay, this is an acidic one. This is common in both DNA and RNA. Now let us discuss the structure of nucleoside, nucleotide, and what is nucleic acid. Alright. What is the pentose sugar part in DNA and RNA? Dear student, I told you the pentose sugar is common in DNA and RNA, but the structures are different and the components are different. Let us study what is the pentose sugar. What are the pentose sugar part in DNA and RNA? Dear all, DNA and RNA, what is the name of pentose sugar? The pentose sugar in RNA is known as beta-D ribose. Beta-D ribose. Beta-D ribose is the pentose sugar of RNA, and beta-D 2-deoxyribose. Beta-D 2-deoxyribose is known as the pentose sugar of which one? DNA. Clear all? Let us draw the shape of the molecule, then it will be clear the name. Dear student, beta-D ribose, the structure is like this. This is the pentose sugar part of RNA. Let me draw the shape. Here we have CH2OH, hydrogen. Both side hydrogen, hydrogen, OH, OH. Okay, CH2OH, hydrogen, HOH, HOH. Here opposite OHH. This structure you try to remember. CH2OH, HOH, HOH, OHH. This is the compound called beta-D ribose, and this is the pentose sugar part of which one? RNA. You see what? There is a small difference in the pentose sugar of RNA and DNA. RNA beta-D ribose. What about DNA? Beta-D 2-deoxyribose, which means second position deoxy, no oxygen. No oxygen. Dear all of you, what are the numbers? Let me write the number of carbon in a pentose sugar. 5 carbon, right? So number 1, 2, 3, 4, 5. These are the 5 carbon in the pentose sugar. So let me draw the same structure here. Same structure here, the pentose sugar. Dear all, this is the pentose sugar. And what we have, this is the number. Numbers we already said. This is the number 1, number 2, number 3, number 4, number 5. Okay. And the species are, here we have CH2OH, H, HOH. Second position deoxy, which means there is no oxygen, right? So you can see second position, there is an oxygen, right? Its oxygen is not there in the DNA. So we can write here hydrogen, here also hydrogen. Okay, so what about here? Same OH. This is the structure of the pentose sugar part of DNA and RNA. Once again, all of you, the pentose sugar of RNA is called beta D ribose. The pentose sugar of DNA is called beta D 2-deoxyribose, which means second position there is no oxygen. Clear? So this is the difference in the pentose sugar of DNA and RNA. Now we can go to nitrogenous base. Nitrogenous base means nitrogen containing one. Dear all, total we have 5 bases. You may study it in biology. The 5 bases are adenine, thymine, guanine, cytosine and uracil. These are the 5 bases, nitrogenous bases. Adenine, thymine, guanine, cytosine, uracil. Right? So which all bases are present in RNA and which all bases are present in DNA? Dear student, DNA contain the bases ATGC. ATGC. What are ATGC? Adenine, thymine, guanine and cytosine. Whereas RNA which contain AUGC. Adenine, uracil, guanine and cytosine. So what is the major difference between adenine and the DNA bases and RNA bases? Dear all, DNA which contain thymine, instead of that RNA having uracil. That is the only difference. So DNA contain ATGC and RNA contain AUGC. Clear all? Okay. So bases also different. So they will ask you the questions like which all bases are common in DNA and RNA. Which all bases are common in DNA and RNA? A, G, C are common. So which is the base only present in DNA? Thymine. Which is the base only present in RNA? Uracil. Clear? So such kind of differentiating question. Now I told you phosphoric acid which is common in both. So this is the major difference between DNA and RNA component. So DNA and RNA on hydrolysis, you will get 3 components: pentose sugar, nitrogen bases and phosphoric acid. Clear all? Now we can prepare what is the structure of this DNA, RNA, everything. We can discuss detail. Dear all of you, DNA and RNA, we call it as nucleic acids, right? So we have nucleic acids, okay? We have nucleic acid. Nucleic acid. Nucleic acid. Nucleic acid which means that is our DNA or RNA. If you want to prepare nucleic acid, we can prepare first of all nucleotide. Nucleotide. So dear all, many number of nucleotides are combining together, you will get nucleic acid. What's the point? N number of nucleotides are combining together and we are getting a big chain of which one? Nucleic acid. So if they ask the question like, what is the simple unit or the monomer? Monomer means a single unit of the polymer nucleic acid. Yesterday, nucleic acid is a big chain. Nucleotides are single species. So this is called a monomer and this is called polymer. So they will ask you what is the monomer of or the simple molecule of nucleic acids. Your answer is what? Nucleotide. Many number of nucleotides are combining together, we will get a nucleic acid. Right? So we can prepare. How can we prepare nucleotide? Dear student, if you want nucleotide, we need the Nucleoside. What it is called? Nucleoside. So nucleoside, from nucleoside you can prepare nucleotide. From nucleotide you can prepare nucleic acid. This is the combination. Now let us discuss what is these three components. So let me discuss about the initial compound nucleoside. Dear student, nucleoside mean a pentose sugar with base. Which base? Nitrogen containing base. Let me take the pentose sugar. Suppose you know the pentose sugar of DNA. What is the pentose sugar of DNA called? Beta D, right? Beta D 2-deoxyribose. Let me draw the shape of that compound, dear all. This is the pentose sugar, right? This is the pentose sugar. What are the components? Do you remember in DNA? Here we have CH2OH, correct? Here hydrogen, OH. This is the second position. Here hydrogen, right? Yes, because there is no oxygen in second position in DNA. And this is OH and H. Alright. And the number of carbon, you know, this is number 1, number 2, number 3, number 4 and number 5. Alright. And dear all, listen carefully. This is the pentose sugar part of DNA. In the first position, remove the OH group. And that position is occupied by the nitrogen containing base. This system is known as nucleoside. So what are the main components in a nucleoside? The pentose sugar with the base. The pentose sugar with the base is known as nucleoside. Can you tell me, dear student, it's very important the position where base is connected. In a nucleoside, the base connected to the first carbon. See, the base connected to the first carbon of the pentose sugar. Yes or no? The base connected to the first carbon of the pentose sugar. This is called nucleoside. Hope you clear? Nucleoside mean pentose sugar with a base. Where is the base present? On the first carbon. Finish? Right. Now we can prepare. So our nucleoside is ready. We need nucleotide, right? Nucleotide means they contain pentose sugar with a base, nitrogen contained base with our phosphoric acid. Alright. So nucleoside which contain pentose sugar base only. Nucleotide contain pentose sugar, base and phosphoric acid. So can I call nucleotide which contain nucleoside with the phosphoric acid? Because nucleoside mean pentose sugar and base, right? These two are nucleoside with the phosphoric acid. Okay, so let me take the nucleoside first of all. Let me take the nucleoside once again, everyone. See all of you, nucleoside, one of the nucleoside. Let me take the what we prepared here, the same nucleoside. Let me take it. What are the system you have? Here you have CH2OH, right? Just let me write the CH2OH like this, CH2OHH. Here we have HOH. Here we have HH. And the first position, it is connected by what? First position is connected by a nitrogen containing base. And here we have hydrogen. This is the pentose sugar with a base. This is called what? Nucleoside. Shall we prepare nucleotide? Yes. This is the nucleoside. Number, please, number 1, number 2, number 3, number 4, number 5. Pentose sugar ready, base is ready, everything is set. Now let us invite our phosphoric acid. What is the formula of phosphoric acid? H3PO4. The structure also we discussed. Hope you remember. What is the structure, everyone? We can draw like this: P double bond O, right? P double bond O. We have OH, 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 right? P double bond O, OH, OH, OH. This is called H3PO4, called phosphoric acid. It's an acid. Okay. When phosphoric acid, hope you clear why I noted the phosphoric acid at this end, because the phosphoric acid is reacting on 5th carbon. Here the phosphoric acid is going to connect on 5th carbon, dear all. What is happening? See all of you, What is your fifth carbon? CH2OH. From your fifth carbon, take the OH group, and from your phosphoric acid, take hydrogen, because that it is an acid, right? It can donate H plus ion. So this hydrogen and OH together, eliminated as what? Water. So H2O is eliminated and they will make a connection here. Let me draw here itself, all of you. What is the connection? Let me remove the area. Okay. Let me remove the area. This is what we have here. One oxygen left, and here it is making a connection. That water part is removed and they will make a connection like CH2O POH OOH. Clear, everyone? This is the one. This compound is known as what? Nucleotide. So what are the main components in nucleotide? Once again, pentose sugar, base. Where is base present? On first carbon. Phosphoric acid. Where is the phosphoric acid present, dear all of you? Phosphoric acid is present on the fifth position of the pentose sugar. Base present on the first position, and phosphoric acid present on the fifth position. Hope you're clear what is the difference between nucleoside and nucleotide. Okay, nucleoside contain pentose sugar and base. Nucleotide contain pentose sugar, base, which means nucleoside, along with the phosphoric acid at the fifth position. Now we can prepare nucleic acid, dear all of you. That is our last concept. Dear student, nucleic acids, I told you, it is preparing from nucleotide. Many number of nucleotides are combining together and we are getting nucleic acid. There is no space to draw all the structures. Let me take two nucleotides, and where is the connection happening? Let us discuss detail. Okay, so let me take one of the nucleotide. I need two nucleotide like this. So I'm taking here a nucleotide. All of you listen carefully. The upper one, let me take it. This is one nucleotide structure. What all things you have, dear all of you? There is a base, hydrogen, here hydrogen, OH, hydrogen. What do you have here? CH2, all of you, CH2O, P, OH, double bond O. This is one nucleotide, right? Into sugar, base, yes. Let me take another nucleotide. Here we have base, hydrogen. Then here hydrogen, OH, H, and then CH2. Now carefully listen how we are drawing this fifth carbon position, okay? CH2O, right? CH2O, P, it is connected to what? One OH group, double bond O, one OH group, alright? This is the one we have, right? Okay. So this is one nucleotide, this is another nucleotide. How they can join together, dear all of you? Hope you clear why we noted these two closer. Dear all, in this case also water is eliminated. From this one alcohol, and from the acid part take this hydrogen. So water is eliminated. So let me remove those water molecule. What is left in the system? Try to understand, all of you. There is an oxygen connection. It is only through a oxygen connection. That's what we have, right? This is the bonding happening in between two nucleotides. So can you tell me, this is one nucleotide, another nucleotide, which all carbons are connecting between two nucleotide connection? Dear all, here we can able to understand. Let me tell you the numbers of each carbon. You know, this is number 1, 2, 3, 4, 5. Here 1, 2, 3, 4, 5. So which all carbons are connecting in a nucleotide? In a nucleotide, you can see the fifth carbon of one nucleotide and the third carbon of the other nucleotide. They are making a connection. Therefore, when you observing the DNA structure, you may study that DNA is a double helical structure, right? DNA is a double helical structure, And RNA is a single-stranded one. It's a single-stranded one, right? So in DNA, one of the strand, we will write it as what? 3 dash, 5 dash. Other one we will write 5 dash, 3 dash. Yes or no? So one strand is 3 dash and 5 dash. What is that? Here 3, you can write it as 3 dash and 5 dash. The other strand will be the complementary one. It will be 5 dash and 3 dash. But RNA is just a single stranded one. So this is the structure formation of which one? DNA and RNA. So like this, a big chain will form, right? Like this. This is nucleoside. So many nucleotides are joined together. So one helical structure, one more helical structure. So it will be double helical. That is the structure of DNA. If it's just one helical, one strand is there, that is called RNA. So DNA is a double stranded one and RNA is a single stranded one. So the connection is clear. Why 5 dash, 3 dash and 3 dash, 5 dash? Here 3 and 5 dash. This is one strand. The other strand is just a complementary one. The 5 dash, 3 dash opposite will be there. Okay. So nucleoside. What is that? What are the main components? What is nucleotide formed from nucleoside? How nucleotides converted to nucleic acids? Hope you clear the connection or where is the bonding happening? Yes or no? So this is one of the major important concept from the chapter biomolecule. They will ask you even for your competitive examination, what all things are in nucleoside, which all things in nucleotide, what is a nucleic acid, which all bonds are connecting, where is the base present, where is the phosphoric acid present, etc. So clear all of you? Understood? So thank you all.